Welcome friends, myself I am Yoganandam, biology lecturer from Edall Academy YouTube channel. In today's class, we are going to discuss one of the important topic from 11th standard biology. The topic is biomolecules. This is one of the important topic for your board examination as well as for your NEET examination. So try to remember the chemicals or molecules present in all living organisms are called as biomolecules. The most amazing thing about a living system is that it is composed of non-living atoms and molecules. There is a wide diversity in living organisms in our biosphere. Now a question is that arises in our mind is, are all living organisms are made up of the same chemicals, that means elements and compounds? If we perform an analysis on a plant tissue, animal tissue or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen and several others and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of Earth's crust as an example of non-living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in the sample of the earth crust are also present in sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other element is higher in any living organism than earth crust. Refer the tabular column. It is very clearly given. Oxygen is the most abundant element in the living system. See the tabular column: hydrogen, carbon, oxygen, their percentage in earth crust as well as in human body is given very clearly in the tabular column, as well as sulfur, sodium, calcium, magnesium, silicon, that also given. Try to remember what is the percentage which is present in earth crust and as well as in human body. It is given very clearly. Then let us go for how to analyze chemical composition, that means chemical analysis. What type of organic compounds are found in living organisms? To get an answer, one has to perform a chemical analysis. Any living tissue, that may be a vegetable or piece of liver tissue etc., is taken and grind it in trichloroacetic acid. Using a mortar and a pestle, we obtain a thick slurry. If we to strain this through a cheesecloth or a cotton, we would obtain two fractions. One is called the filtrate, or more technically the acid soluble pool. The second one is the retentate, or the acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. Inorganic compounds like sulfate, phosphate etc. are also seen in the acid soluble fraction. Analytical techniques, when applied to the compound, give us an idea of the molecular formula and the probable structure of the compound. All the carbon compounds that we get from living tissue can be called as biomolecules. Try to remember, all the carbon compounds that we get from living tissue can be called as biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? Yeah, slightly different, but destructive experiments has to be done. One, weight is a small amount of a living tissue, say a leaf or liver, and this is called wet weight, and dry it. All the water evaporates, the remaining material gives dry weight. Now if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form in the form of carbon dioxide, water vapor and are removed. What is remaining is called as ash. This ash contains inorganic elements like calcium, magnesium, etc. Therefore, the elemental analysis gives elemental composition of living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compounds gives an idea of the kinds of organic and inorganic constituents present in living tissues. So you try to remember which are the things we have discussed. Then let us discuss about biomolecules. The molecules, there is one fraction common to all those compounds found in the acid soluble pool. They have the molecular weight ranging from 18 to around 800 daltons approximately, or less than 1000 daltons. Such kind of molecules are called as micromolecules. Micromolecules may be organic or inorganic and includes water, minerals, amino acids, sugar, nucleotides and gases. Try to remember, it is very clearly given in the tabular column. What are the different types of micromolecules given? Refer the flow chart. Glucose, ribose, amino acids, nitrogen bases, nucleotides, nucleosides. These are all some common examples. So these molecules, molecular weight ranging from 18 to 800 dalton, present in acid soluble pool. Macromolecules are always organic. The acid insoluble fraction has only 4 types of organic compounds such as proteins, nucleic acids, polysaccharides and lipids. Macromolecules molecular weight range is 10,000 dalton or above, present in acid insoluble pool. Try to remember what are the 4 examples which are given for macromolecules: proteins, nucleic acid, polysaccharide and lipids. Lipids exception. These classes of compounds with the exception of lipids have molecular weight in the range of 10,000 daltons and above. For this very reason, biomolecules, the chemical compound found in living organisms, are 2 types. One of those which have the molecular weight less than 1000 dalton and are usually referred to as micromolecules or simple biomolecules, while those which are found in acid insoluble fraction are called as macromolecules or biomacromolecules. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Except a lipid or other macromolecules are formed by polymerization or condensation of monomers. Hence, these are called polymers. Proteins are polymers of different amino acids. Polysaccharides are otherwise called as carbohydrate or polymers of monosaccharides. Nucleic acids are the polymers of nucleotides. Let us discuss about micromolecules followed by macromolecules. From micromolecules, first we are going to discuss about amino acids. It is one of the example for micromolecule. Amino acids are organic compounds containing an amino group and an acidic group as a substituent on the same carbon. For example, alpha carbon. Hence, they are called alpha amino acids. They are substituted methanes. These are 4 substituent group occupying the 4 valency positions. These are hydrogen, carboxyl group, amino group and a variable group designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only that are 20 types. The R group in these proteinaceous amino acids could be a hydrogen, and amino acid is called as glycine. A methyl group, that is alanine, hydroxymethyl group, example serine, etc. The chemical and physical properties of amino acids are essentially of the amino, carboxyl and or functional groups. Based on the number of amino and carboxyl groups, there are acidic, example glutamic acid, basic, example lysine, and neutral, example valine. These are the three types of amino acids. Based on the number of amino acids and carboxyl group, there are acidic, basic and neutral. Try to remember with example very clearly given. A particular property of amino acid is the ionizable nature of NH2 and COOH group. Hence, in solutions of different pH, the structure of amino acids undergoes certain changes. In aqueous solution, the carboxyl group can lose a proton and the amino group can accept a proton, giving rise to the dipolar ion called Zwitter ion. Zwitter ion is neutral but contains both positive and negative charges. Refer the diagrammatic representation. At the same time, the amino acids can also be classified as essential amino acids and non-essential amino acids. The essential amino acids are not synthesized by the body and should supply through diet. Example lysine, leucine, isoleucine, tryptophan, etc. Non-essential amino acids can be synthesized by the body. Example glycine, alanine, serine, arginine. So it is another kind of classification that also you try to remember. It is followed by lipids. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to a R group. The R group could be a methyl, that is CH3, or ethyl C2H5, or a higher number of CH2 groups, 1 carbon to 19 carbons. Fatty acids could be saturated, that means without double bond, or unsaturated with one or more double bonds in between CC. Saturated fatty acids do not have double bond in their carbon chain. Example, palmitic acid. The molecular formula is given, refer the diagrammatic representation. Palmitic acid has 16 carbons including carboxyl carbon. In lipids, the number of oxygen atom is always less than the number of carbon atoms. Unsaturated fatty acids have one or more double bonds in their carbon chain. Arachidonic acid has 20 carbon atoms including the carboxyl carbon. Another simple lipid is glycerol, which is trihydroxypropane. Glycerol is a 3-carbon alcohol with 3 OH group as binding sites. Many lipids have both glycerol and fatty acids. These are also called fats and oils based on melting point. Oils have lower melting point, example gingerly oil, and hence remain as oil in winters. Hence, the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides and triglycerides. In this slide, it is very clearly given about triglycerides. One glycerol and one fatty acid together form monoglyceride. One glycerol and 2 fatty acid together form diglycerides. One glycerol and 3 fatty acid together form triglycerides. It is followed by compound lipids. These are the esters of fatty acids and alcohol with additional groups. Example, phospholipids. It is composed of fatty acid, glycerol and phosphate group. They are found in cell membrane. Here one of the example is given for a compound lipid, that is lecithin. Compound lipids followed by derived lipids. These are the products of hydrolysis of simple lipids and compound lipids. One of the example is cholesterol. Another example is sugar. Sugar is always called as carbohydrate. Carbohydrates are polyhydroxyaldehydes or ketones are the compounds which produce such units on hydrolysis. Carbohydrates are also called as saccharides. Some carbohydrates are sweet in taste and called the sugars. Carbohydrates are made up of carbon, hydrogen and oxygen in the ratio of 1:2:1. Examples: glucose and ribose. It is followed by nitrogenous bases, also given under micromolecules. These are the nitrogen containing cyclic compounds found in nucleic acids. These are classified as follows. Nitrogen bases are broadly divided into purines and pyrimidines. Purines, example adenine and guanine. Pyrimidines, example cytosine, thymine and uracil. See the flow chart. Nitrogen bases with diagrammatic representation, it is given very clearly, adenine and uracil. What do you know about nucleosides and nucleotides? Nucleoside, there are 5 types of nucleosides are given. Try to remember. Adenine and sugar together form adenosine nucleoside. Guanine and sugar together form guanosine. Cytosine and sugar together form cytidine. Thymine and sugar thymidine. Uracil and sugar uridine. So you try to remember nitrogen base and sugar without phosphate group. Nucleosides are made up of nitrogenous base and sugar. Nitrogenous base, sugar and phosphate group together form nucleotide. Nucleotide without phosphate group is called as nucleoside. Same like nucleoside, there are 5 types of nucleotides are also given. Try to remember. Adenine, that is nitrogenous base, sugar and phosphate group together form adenylic acid. Guanine, sugar and phosphate together form guanylic acid. Cytosine, sugar and phosphate together form cytidylic acid. Thymine, sugar and phosphate together form thymidyllic acid. Uracil and sugar and phosphate together form uridylic acid. So these are the 5 types of nucleotides, as same like nucleosides. Nucleotides are heterocyclic compounds. Nucleic acids such as DNA and RNA are made up of nucleotides. Nucleotides are made up of nucleosides. So it is followed by primary and secondary metabolites. First we must know what is metabolism. The organic compounds taking part in metabolism is called as metabolites. The most exciting aspect of chemistry deals with the isolating thousands of compounds, small and big, from living organisms, determining their structure and, if possible, synthesizing them. If one were to make a list of biomolecules, such as list would have thousands of organic compounds including amino acids, sugars, etc. We can call these biomolecules as metabolites. In animal tissues, one notices the presence of all such categories of compounds like sugars, amino acids, fat, oil, liquids, nitrogenous bases, nucleosides and nucleotides, which are called primary metabolites. So refer flow chart. However, when one analyzes plant, fungal and microbial cells, one word to see the thousands of compounds other than primary metabolites are called as secondary metabolites. Example: alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices, etc. So you try to remember, while the primary metabolites have identifiable functions and play known roles in normal physiological processes, we do not know at the moment understand the role or functions of all the secondary metabolites in host organisms. However, many of them are useful to human welfare. Example: rubber, drugs, spices, scents, and pigments. Some secondary metabolites have ecological importance also. So you try to remember everything is given in the tabular column. Secondary metabolites, pigments, alkaloids, terpenoids, essential oils, toxins, lectins, drugs, polymeric substances. For all these secondary metabolites, examples are given very clearly. What are the differences between primary metabolites and secondary metabolites? That also try to understand. Primary metabolites have identifiable functions in the physiological process and necessary for life. So while we are talking about secondary metabolites, they are not directly involved in the normal growth, development, or reproduction of organisms. So that is the basic difference between primary metabolites and secondary metabolites. Biomicromolecules followed by biomacromolecules. So you know the differences between micromolecules and macromolecules. A molecular weight in range 10,000 daltons or above are called as macromolecules. Is present in acid insoluble pool. There are 4 examples already we have discussed. Try to remember: protein, nucleic acid, polysaccharide. What about lipid? They are given except lipid. Then why do lipids whose molecular weight do not exceed 800 dalton come under acid insoluble fraction? For example, macromolecular fraction. Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. The cell membrane and other membranes are also broken into pieces and form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid soluble pool represents roughly the cytoplasmic composition. The macromolecules form cytoplasm and organelles become the acid insoluble fraction. Together they represent the entire chemical composition of living tissue or organisms. If we represent the chemical composition of living tissue from abundance point of view and arrange them class-wise, we can observe that water is the most abundant chemical in living organisms. So let us discuss about one by one biomacromolecules. So except lipid, there are 3 examples we are going to discuss under biomacromolecules: proteins, polysaccharides, and nucleic acids. First, let us discuss about proteins. So you all know proteins are polypeptides. They are linear chains of amino acids linked by peptide bonds. Each protein is a polymer of amino acid, as there are 20 types of amino acids. Example: alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. Amino acids can be essential or non-essential. That already we have discussed. Essential amino acids are important for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. The non-essential amino acids are those which our body can make. So you try to remember what are the 2 types, essential and non-essential amino acids. So let us discuss about the structure of proteins. Proteins are heteropolymers containing string of amino acids. In inorganic chemistry, the structure invariably refers to the molecular formulae NaCl, MgCl2, etc. That is sodium chloride, magnesium chloride, etc. Organic chemists always write a 2-dimensional view of the molecules while representing the structure of the molecule. example benzene, naphthalene, etc. Physicists conjure up the three-dimensional views of the molecular structures, while biologists describe the protein structure at four levels. What are the four levels of proteins? Let us discuss one by one. First one is primary structure of protein. The sequence of amino acids, for example, the positional information in a protein, which is the first amino acid, which is second, and so on, is called as primary structure of protein. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called as N-terminal amino acid. The last amino acid is called as C-terminal amino acid. It is followed by secondary structure. The second structure, that is a secondary structure of protein, refers to the shape in which long polypeptide chain can exist. A protein thread does not exist throughout as an extended ridged rod. The thread is folded in the form of a helix, similar to a revolving staircase. Of course, only some portions of the protein thread are arranged in the form of a helix. In proteins, only right-handed helix are observed. Other regions of the protein thread are folded into other forms, is what called as secondary structure. It is followed by tertiary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure. This gives us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities of proteins. Example, myoglobin. So try to remember about the tertiary structure of the protein. This long protein chain is also folded upon itself like a hollow woolen ball. It gives three-dimensional view of protein. It is followed by quaternary. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individuals folded polypeptides or subunits are arranged with respect to each other. Example, linear string of spheres. Spheres arranged one upon each other in the form of a cube or plate, etc. is the architecture of the protein, otherwise called as the quaternary structure of the protein. Adult human hemoglobin consists of, there are 4 units. There are 4 subunits. 2 or these are identical to each other. Hence, 2 subunits of alpha type and 2 subunits of beta type together constitute the human hemoglobin. Try to remember, some proteins are an assembly of more than one polypeptide or subunits. Example, human hemoglobin has 4 subunits, such as 2 alpha subunits and 2 beta subunits. Try to remember, the most abundant protein in the animal world is collagen. At the same time, the most abundant protein in the whole of the biosphere is rubisco. Rubisco means ribulose biphosphate carboxylase oxygenase enzyme. Proteins carry out, there are several functional activities. Proteins carry out many functions in living organisms. Some kind of proteins transport nutrients across cell membrane. Some fight infectious organisms. Some are hormones. Some are enzymes, etc. So try to remember this flow chart. Proteins essential for growth and tissue repair, transport of nutrients across cell membrane. For example, GLUT4 enables glucose transport into cell, act as intracellular ground substance, example collagen, act as antibodies to fight infectious organisms, act as receptors, example receptors of smell, taste, hormones. Some are hormones, example insulin. Some are enzymes, example trypsin. Some are pigments, example hemoglobin, etc. So you try to remember which are the examples given, that everything is important. Structure of protein followed by polysaccharides. The acid insoluble pellet has polysaccharides such as carbohydrates as another class of macromolecules. Polysaccharides are long chains of sugars. They are threads, that means literally a cotton thread containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide. For example, glucose. Homopolysaccharides, starch, glycogen, and cellulose are the polymers of glucose. Starch. Starch is a variant of cellulose and present as a storehouse of energy in plant tissue. Starch forms helical secondary structures. In fact, starch can hold I2 molecule, that is iodine molecule, in the helical portion. The starch iodine is blue in color. Cellulose does not contain complex helix and hence cannot hold iodine. A second example, glycogen. Animals have another variant called as glycogen. It is commonly called as animal starch because its structure is similar to amylopectin. In a polysaccharide chain, it is generally called as glycogen. The right end is called as reducing end and the left end is called as non-reducing end. It has branches. And another example is cellulose. Cellulose is a homopolymer. Plant cell walls are made up of cellulose. Paper made from plant pulp and cotton fiber is made up of cellulosic. And another important example is chitin. There are more complex polysaccharides in nature. They have building blocks, amino sugars, and chemically modified sugars. Example, glucosamine, N-acetyl glucosamine, etc. These polysaccharides are formed by the condensation of two or more types of monosaccharides. For example, exoskeletons of arthropods, for example, have a complex polysaccharide called as chitin. These complex polysaccharides are homopolymers. Then another example is unulin. It is a polymer of fructose. So these are all some examples given for polysaccharides. Try to remember. Starch forms helical secondary structure. Starch holds iodine molecule in the helical portion giving blue color. But cellulose has no complex helix and therefore cannot hold iodine. It is followed by the important example which is given for macromolecule is nucleic acids. Nucleic acids are broadly divided into DNA and RNA. Nucleic acids are heteropolymers of nucleotides. Many nucleotides are linked to form polynucleotide chain. So you try to remember. The other type of macromolecule that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with the polysaccharides and the polypeptides, these comprise the true macromolecular fraction of any living tissue or cell. For nucleic acids, the building block is called as nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is the monosaccharide, and the third is phosphoric acid or phosphate. The heterocyclic compounds in nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are the substituted purines, while the rest are substituted pyrimidines. The sugar found in the polynucleotide is either ribose, that is monosaccharide pentose, or 2-deoxyribose. The nucleic acid containing deoxyribose is called as deoxyribonucleic acid, that is DNA, while that which contains ribose is called as ribonucleic acid, RNA. Adenosine, guanosine, thymidine, uridine, and cytidine are the nucleosides. Adenylate acid, thymidylate acid, guanylic acid, uridylic acid, and cytidylate acid are generally called as nucleotides. This and all already we have discussed under nitrogenous bases from biomicromolecule itself, you try to remember the common type of DNA is Watson and Crick model. It is the secondary structure of DNA. So according to Watson and Crick model of DNA consists of, there are two polynucleotide strands arranged anti-parallelly as a double helix. One full turn as there are 10 steps, that means 10 base pairs. The length of one full turn is 34 Armstrong. In nanometer it is a 3.4. The same time the distance between each step is 3.4 Armstrong. At each step the strand turns 36 degrees Celsius, that means 360 degrees for a one full turn. In DNA, a nucleotide consists of nitrogen base, deoxyribose sugar and a phosphate group. The backbone of DNA strand is formed by the sugar, phosphate, sugar chain. That is, you try to remember, the backbone of DNA strand is formed by sugar, phosphate and sugar chain. Steps are formed of nitrogen base pairs. Nitrogen bases include adenine, guanine, thymine and cytosine. Uracil is absent in DNA. Try to remember Chagov base pair rule. According to Chagov base pair rule, adenine always pair with thymine. In between adenine and thymine, there are two hydrogen bonds are present. Guanine always pair with cytosine. In between guanine and cytosine, there are three hydrogen bonds are present. A phosphate molecule links the third carbon atom of one sugar of one nucleotide to the fifth carbon of the sugar of the succeeding nucleotide. There is an ester bond between phosphate group and OH group of sugar. As there is one such ester bond on either side, it is generally called as phosphodiester bond. The bond between sugar and nitrogenous base is called as N-glycosidic bond. So this and all, once again we are going to study from 12th standard, molecular basis of inheritance. First we are going to discuss about dynamic state or body constituents concept of metabolism. What we have learned till now is that is living organisms, be it is a simple bacterial cell, a protozoan, a plant or an animal, contain thousands of organic compounds. These compounds or biomolecules are present in certain concentrations that may be expressed in cell mass or cell liter, etc. One of the greatest discoveries ever made was the observation that all these biomolecules have a turnover. This means that they are constantly being changed into some other biomolecules and also made from some other biomolecules. This breaking and making is through chemical reactions constantly occurring in living organisms. Together, all these chemical reactions are called as metabolism. Try to remember which topic we are discussing: metabolism. Each of the metabolic reactions results in the transformation of biomolecules. A few examples for such metabolic transformations are removal of carbon dioxide from amino acids, making an amino acid into an amine, removal of amino group in nucleotide base, hydrolysis of glycosidic bond in a disaccharide, etc. We can list tens and thousands of such examples. Majority of these metabolic reactions do not occur in isolation but are always linked to some other reactions. In other words, metabolites are converted into each other in a series of linked reactions called metabolic pathways. These metabolic pathways are similar to the automobile traffic in a city. These pathways are either linear or circular. These pathways crisscross each other. For example, there is a traffic junctions. Flow of metabolites through metabolic pathway has a definite rate and direction like automobile traffic. This metabolite flow is called as dynamic state of body constituents. What is the most important is that this interlinked metabolic traffic is very smooth and without a single reported mishap for healthy conditions. Another feature of this metabolic reaction is that every chemical reaction is called as catalyzed reaction. There is no uncatalyzed metabolic conversion in living systems. Even carbon dioxide dissolving in water, a physical process, is a catalyzed reaction in living systems. The catalyst which hasten the rate of a given metabolic conversion are also proteins. These proteins with catalytic power are named enzymes. It is followed by metabolic basis for living. Metabolic pathways can lead to a more complex structure from a simpler structure. For example, acetic acid become cholesterol or lead to a simpler structure from a complex structure. For example, glucose becomes lactic acid in our skeletal muscle. The former case are called biosynthetic pathway or anabolic pathways. The later constitute degradation and hence called catabolic pathways. Anabolic pathways, as expected, consume energy. Assembly of proteins from amino acids requires energy input. On the other hand, catabolic pathways lead to the release of energy. For example, when glucose is degraded to lactic acid in our skeletal muscle, energy is liberated. This metabolic pathway from glucose to lactic acid, which occurs in 10 metabolic steps, it is generally called as glycolysis. Living organisms have learned to trap this energy liberated during degradation and store it in the form of chemical bonds. Enzyme made available in catabolic reaction is immediately stored in ATP. As and when needed, this bond energy is utilized for biosynthetic, osmotic, and mechanical work that we perform. The most important form of energy currency in living system is the bond energy in a chemical called adenosine triphosphate. It is generally called as ATP. How do living organisms derive their energy? What strategies have they evolved? How do they store this energy and in what form? How do they convert this energy into work? You will study and understand all these under a sub-discipline called as bioenergetics. So it is followed by the living state. As this level, you must understand that the tens and thousands of chemical compounds in a living organism, otherwise called as metabolites or biomolecules, are present at concentrations characteristics of each of them. For example, the blood concentration of glucose in a normal healthy individual is 4.5 to 5 mm, while that of hormones would be nanograms per ml. The most important fact of biological system is that all living organisms exist in a steady state characterized by concentrations of each of these biomolecules. These are biomolecules are in a metabolic flux. Any chemical or physical process moves spontaneously to equilibrium. The steady state is non-equilibrium state. One should remember from physics and systems at equilibrium cannot perform work. All living organisms work continuously. They cannot afford to reach equilibrium. Hence, the living state is a non-equilibrium steady state. To be able to perform work, living process is a constant effort to prevent falling into equilibrium. This is achieved by the energy input. Metabolism provides a mechanism for the production of energy. Hence, the living state and metabolism are synonymous. Without metabolism, there cannot be a living state. The living state followed by enzymes. Almost all enzymes are proteins. There are some nucleic acids that behave like enzymes. They are called ribozymes. An enzyme, like any protein, has a primary structure. For example, amino acid sequence of protein. An enzyme, like any protein, has secondary and the tertiary structure. At the tertiary structure, the backbone of the protein chain fold upon itself. The chain crisscross itself and ends many services or pockets are made. One such pocket is called as active site. An active site of an enzyme is a service or pocket into which the substrate fits. Thus, enzyme, though their active site catalyze reactions at high rate. Enzyme catalysts differ from inorganic catalysts in many ways, but one major difference need mention. Inorganic catalysts work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperatures that may be above 40 degrees Celsius. However, enzymes isolated from organisms who normally live under extremely high temperatures. For example, hot winds and sulfur springs are stable and retain their catalytic power even at high temperatures up to 80 degree to 90 degrees Celsius. Thermal stability is thus an important quality of such enzymes isolated from thermophilic organisms. So it is followed by chemical reactions. The chemical compounds undergoes there are two types of changes. A physical change simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter. When ice melts into water or when water becomes a vapor, these are physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called as chemical reaction. For example, here one of the equation is given, it is barium with hydrogen sulfide. It gives barium sulfate and water. So that is one of the reaction is given. It is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of physical or chemical process refers to the amount of product formed per unit time. It can be expressed as delta P by delta T. So refer the equation and rate which is given with the help of symbols. Rate can also be called velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature among other factors. A general rule of thumb is that rate doubles or decreases by half for every 10 degrees Celsius change in either direction. Catalyzed reactions proceed at rates vastly higher than that of uncatalyzed ones. When enzyme catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reactions. For example, carbon dioxide with water, the presence of carbonic anhydrase gives carbonic acid. So refer the equation, it is given very clearly. In the absence of any enzyme, this reaction is very slow with about 200 molecules of carbonic acid being formed in an hour. Per hour, how much amount of carbonic acid formed, that is given very clearly. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakhs molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzyme is incredible indeed. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzymes, is called metabolic pathway. For example, glucose converted into 2 molecules of pyruvic acid. It is generally called as glycolysis. Refer the equation. It is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzymes catalyzed metabolic reactions. Refer the steps, that is given very clearly with the help of flow chart. Well, you are seeing how does the glucose is converted into pyruvic acid. It is very clearly given with the help of enzymes. At this stage, you should know that in glycolytic pathway with one or two additional reactions gives rise to a variety of metabolic end products. In our skeletal muscle, under anaerobic conditions, lactic acid is formed. Under normal aerobic conditions, pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol. It is called as alcohol. Hence, in different conditions, different products are possible. So, you try to remember. It is followed by how do enzymes bring out such high rates of chemical conversions? Let us discuss. The chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is called substrate. Hence, enzymes, that is proteins with three-dimensional structures including an active site, convert a substrate, it is generally denoted as S, into product P. Symbolically, this can be depicted, refer the flow chart. It is now understood that the substrate denoted as ES has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of an enzyme substrate. It is denoted as ES complex. E stands for enzyme. The complex formation is a transient phenomenon during the state where substrate is bound to the enzyme active site. A new structure of the substrate called as transition state structure is formed. Very soon after the expected bond breaking or making is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into the structure of product. The pathway of this transformation must go through the so-called transition state structure. There could be many more altered structural states between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy status of the molecular or the structure. The y-axis represents the potential energy content. You would notice two things: the energy level difference between S and P. If P is at a lower level than S, the reaction is an exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or a spontaneous reaction or an endothermic or energy requiring reactions, the S has to go through a much higher energy state of the transition state. The difference in an average energy content of S from that of this transition state is called as activation of energy. Chemical reactions require energy for activation. Enzymes eventually bring down this energy barrier. It is generally called as activation energy, making the transition of ES to P more easy. It is a substrate to product. So you try to understand how does the high rates of chemical conversion takes place with the help of enzymes. It is followed by nature of enzyme action. Each enzyme denoted as E as a substrate, that is S binding site in its molecule, so that a high reactive enzyme substrate complex is produced. This complex is short-lived and dissociates into its products P and the unchanged enzyme with an intermediate formation of the enzyme product complex that denoted as EP. The formation of the ES complex is essential for catalysis. Try to remember, enzyme bind with substrate to form enzyme substrate. Then enzyme substrate that can be converted into enzyme product. Then enzymes and product both are released from one another. The catalytic cycle of an enzyme action can be described in the following steps. First, the substrate bind to the active site of the enzyme, fitting into the active site. The binding of substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. Then the active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme product complex is formed. Then the enzyme released the products of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. These are the steps given. How does the enzymes carry out the functional activities? That means the nature of enzyme action given very clearly, you try to understand. It is followed by factors. Some factors are affecting enzyme activity. Let us discuss about what are the factors which are affecting enzyme activity. The activity of an enzyme can be affected by the change in the conditions which can alter the tertiary structure of the protein. These include temperature, pH, change in substrate concentration, or binding of specific chemicals that regulate its activity. Let us discuss one by one. The first one is temperature and pH. Enzymes generally function in a narrow range of temperature and pH. pH means percentage of hydrogen ion concentration. Each enzyme shows its highest activity at a particular temperature and pH called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. The second factor is concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, is generally called as Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecule to bind with the additional substrate molecule. As Km indicates the substrate concentration at which reaction attains half of its maximum velocity. Km velocity of the enzyme is substrate concentration at half Vmax. The curve is hyperbolic. Then the third factor is inhibitors. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzyme's activity, the process is called inhibition and the chemical is called as inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. Due to its close structure, similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind and as a result, the enzyme action declines. Example, inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. You try to remember what are the factors which are affecting the enzyme activity: temperature and pH, concentration of substrate, inhibitors. Okay, these are the factors affecting the enzyme activities. Factors affecting enzyme activity followed by Classification and nomenclature of enzymes. There are thousands of enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into different groups based on the type of reactions they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named according by the 4-digit numbers. First one is oxidoreductases. It is service called as dehydrogenases. Enzymes with catalyze oxidoreduction between two substrates, that is S and S, or transfer of electrons. Example, substrate reduced and substrate oxidized gives substrate oxidized and reduced. And another enzyme is transferases. Enzymes catalyzing a transfer of a group is generally called as G other than a hydrogen between a pair of substrate. It is denoted as S and S. Example, how does the enzyme catalyzes? That is very clearly given in the form of equation. You go through the equation. It is followed by hydrolysis. Enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic is in between carbon and carbon, carbon and halide, or P and N bonds. It is followed by lyases. Enzymes that catalyze a removal of groups from substrates by mechanism other than hydrolysis, leaving double bonds. So refer the equation, it is very clearly given. Then the next one is isomerases. It includes all enzymes catalyzing interconversion of optical, geometric or positional isomers. Then the sixth one is ligases. Enzymes catalyzing the linking together of two compounds. Enzymes which catalyze joining of CO or CS or CN or PO etc. bonds. So you try to remember what are the 6 classes, that everything is very clearly given one by one. Try to remember once again. Oxidoreductases, it is service called as dehydrogenases. Second type is transferases. Third is hydrolysis. Fourth type is lyases. Fifth one is isomerases. Sixth type is ligases. Okay. It is followed by cofactor. This is the last topic from biomolecules. Okay, let us continue the topic. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called as cofactors are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called as apoenzyme. 3 kinds of cofactors may be identified, such as prosthetic groups, coenzymes, and metal ions. First one is prosthetic groups. Prosthetic groups are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is the prosthetic group and it is a part of the active site of the enzyme. It is followed by coenzymes. Coenzymes are also organic compounds, but their association with their apoenzyme is only transient, usually occurring during the course of catalysis. Furthermore, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Example, coenzyme nicotinamide adenine dinucleotide, it is generally called as NAD, and NADP contain the vitamin niacin. Every coenzyme is a cofactor, but every cofactor is not a coenzyme. Try to understand. So which are the things we have discussed under coenzymes? It is followed by metal ions. A number of enzymes required metal ions for their activity, which form coordination bonds with side chains at the active site and at the same time form one or more coordination bonds with the substrate. For example, zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Try to remember, one of the example is given. Zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. So we have completed the topic biomolecules.
லைக் பண்ணுங்கள், ஷேர் பண்ணுங்கள், கமெண்ட் பண்ணுங்கள், சப்ஸ்கிரைப் பண்ணுங்கள். நன்றி, வணக்கம்.